because the normal function is involved in lots of different biological processes, it's really important to understand all of the biological processes where all of these tumor suppressor genes and oncogenes are involved in. So here I'm talking mostly about genetic changes in DNA alteration or translocations and things like that. But you have to relate all of the genetic changes to the processes in the cells, which are cell apoptosis, cell cycle, or cell signaling, right? So another type of modification that can happen at the epigenetic level are histone modification. And histone modification actually have an impact on post-translational modification. So it can be acetylation, or it can be methylation, so you have methylation of your promoter, or you have ubiquitination or smolation. And the effect on the expression of DNA is at the level of transcription, but you can also have an effect on repair, on replication, or on recombination. So to touch on a bit on hereditary cancers, there's more than 20 different hereditary cancer syndromes. Here is an example of some of these dominantly inherited cancer syndromes. So P53, again, one of these really major tumor suppressor genes, is found mutated at the German level, and it causes a syndrome which is called lymphomany. So in lymphomany, you have lots of different types of cancers, and you can have some of these bone cancers, osteosarcoma. I'll give you an example actually of one of these cancer syndromes, which is familial adenomatous polyposis. And in this syndrome, the gene mutated is called APC, and is also involved in non-inherited cancer. I'll explain a bit more later. I'm also going to develop a bit breast and ovarian cancer with BRCA1 and BRCA2, or also hereditary non-polyposis colorectal cancer, which is when I spoke about the mismatched repair genes with microsatellite instability. So as an example, colon cancer. So there's two different types of hereditary colon cancer. Actually, there is three of them, but the last one is so rare that I'm not going to talk about it. What you have also to remember, and I've said that already in the beginning, hereditary cancer it's rare. It's less than 10% of cancer which are hereditary. Most of the cancer, they are sporadic and they require somatic mutations. So less than 1% of colon cancer have a mutation in a gene called APC, and that causes familial adenomatous polyposis. And the other example of hereditary colon cancer is HNPCC, where it represents around 3–5% of all colon cancer. And there is a defect in the mismatch repair genes that I will explain later. So that's a nasty figure, actually, a nasty pictures of FAP. It's a rare autosomal dominant trait. And the mutation is in a gene called APC. It's a tumor suppressor gene, APC, and it's on chromosome 5, on 5q22. And as you can see in the pictures, you already actually have thousands of polyps. And these polyps, at this stage, they're not malignant, they are benign. But then they're going to acquire more mutations, and then they will become malignant. And it's a childhood onset because, as you can see, you already had a mutation in APC that had created all of these thousands of polyps. And it only needs a mutation in the other, in a couple of 2 or more genes, and then it becomes malignant. And so that's why hereditary cancer, usually they have an early age of onset compared to a sporadic cancer. Another example is HNPCC. It's also called Lynch syndrome. So if you do a bit of studies and if you're interested in colon cancer, HNPCC is also called Lynch syndrome, and it's called Lynch because it's been discovered by Henry. In that case, you have less polyps. It's less than 100 polyps. And there's a high risk of colon cancer in people having a mutation in these mismatched repair genes. But you can also have a higher incidence of other cancer, like endometrial cancer, ovary cancer, or intestine. It's an autosomal dominant inheritance, and the incidence is like 1 in 25,000. And it represents 3 to 10% of all colorectal carcinomas. And again, because it's in the hereditary context, it's an early age onset disease. It happens around 40, 50 years. When usually sporadic colon cancer arises after 60 or 70 years old. But if you had already acquired mutations, then you develop cancer earlier. So in HNPCC, you have different genes which are found to be mutated, and they are DNA repair genes. And these genes are NSH2 or NLH1 or sometimes NSH6. The most frequent is in mutation reported in HPNCC is NLH1. You have 50% of this type of cancer who have a mutation in NLH1. And usually the mutation reported for this type of genes are at the DNA level. So it's a change of nucleotide, or a small deletion or a small insertion. And it's very rare to have big rearrangement of deletions. You can have also a mutation in MSH2, which represents 40% of all mutations which have been described for HNPCC. So here's a bit of I think that's quite an important slide, because it shows you the progression from the normal epithelium to colon cancer. And it gives here 3 different types of examples. On the top line, you have sporadic cancer, sporadic colon cancer. In the middle, you have the FLP, the one where I showed you there's thousands of polyps because there's a mutation in the gene called APC. And at the bottom, you have the other example with HNPCC. So if you remember the previous slide, HNPCC is developed in adults, around 40 years old, 50 years old. FLP is more like kids, so it's a really early onset. And then sporadic colon cancer is later on when it's 60, 70, 75 years old. And the reason why is because when it's in the sporadic context, you need to acquire all of these different genetic changes in the cells before it becomes really malignant. So you see at the beginning, you have a mutation in APC, and you only have like early adenoma, so it's a benign tumor. And then it becomes, you acquire another mutation in a KRAS gene, which is one of these genes which are involved in cell signaling. And then this becomes an intermediate adenoma. And then it acquires another couple of mutations in some other tumor suppressor genes, like P53, and then it becomes fully malignant, and it becomes a carcinoma. And when you have a carcinoma, you also have some more genetic mutations, and then it becomes metastatic, and then it's going to spread and localize to some other organs. So you see, it takes a long time before all of these genetic changes take place before the tumors become fully malignant and metastasize. But if you had already acquired a mutation, then it's quicker. So that's the reason why in FLP you have a mutation in APC, and very quickly it becomes a late adenoma. You acquire another mutation in P53 and it becomes fully malignant and metastasized. And in HNPCC, it's slightly the same, but it's not a mutation actually in APC, it's in one of these mismatched repair genes. So it's DNA repair, so it takes a bit longer before you acquire mutations. But then eventually, because you have this DNA repair which is defective, you acquire more mutation in some other genes and proteins or tumor suppressor genes, and then the tumor becomes polyvalent. Hereditary breast cancer, again, it's an autosomal dominant inheritance. It's an earlier age of onset of breast cancer, usually when it's in the context of predisposition to breast cancer. Women have cancer before the age of 45. Compared to sporadic breast cancer, when you have hereditary breast cancer, it affects both breasts. And you can also develop more tumors, this type of hereditary breast cancer. So hereditary breast cancer is when there is a clear evidence of the transmission of mutated genes. You can also have familial breast cancer, where there's a family history of one or more first or second degree relative with breast cancer, but there's not necessarily a transmission of the cancer genes. And in more than 70% of the case, you have sporadic breast cancer, where there is no family history of breast cancer. You can have, and this is what we call phenocopies, you can have in a family a breast cancer in one of your relatives, but actually it just had occurred by chance, so it's not due to hereditary. And it's the same thing when you're in a familial context. You can have obviously a transmission of mutated genes like BRCA1 or BRCA2, but you can have also sporadic breast cancer, and that is called phenocopy, because this breast cancer hasn't been due to a mutation in one of these cancer genes. So what are the genes involved in hereditary breast cancer? So you have BRCA1 and BRCA2. You have also P53, and that's the deformity syndrome, where you have breast cancer and also bone tumors. You can have also mutation in another gene. I haven't talked about it, but it's STK, which is another hereditary cancer syndrome, the third stages. You can have also a mutation in the tumor suppressor gene called P10, and the syndrome is known as Gordon disease. You can have also mutation in a mode called CDH1, and the other name for CDH1 is ecadherin. And I think when you're going to see the epithelial to mesenchymal transition that will be done by Caroline later, you will see that mutated gene is CDH1. So you'll see that CDH1 can be associated with hereditary cancer, but also non-hereditary cancer. BRCA1 is an important component of pathways that regulates, and that goes back to the discussion we had before. But BRCA1 is important DNA repair in cell cycle progression, in methylation and transcriptional regulation. So it's really important. That's why when you have a mutation in one of these genes, it dysregulates the whole normal cells. You can find lots of different types of BRCA1 mutations. BRCA1 mutations are not found in sporadic breast cancer. It's only in the hereditary context. And you have LOH, which is what I described before. You have one allele which is mutated and transmitted through the generation, and you have loss of the wild type allele in the breast tumor cells. That's in the somatic breast cells. There are very rare mutations found in sporadic cancer, and actually it's not found in breast cancer, it's found in ovarian cancer. But BRCA1, interestingly, can also be involved in sporadic breast cancer, but it won't be by classic mutation, it will be by epigenetics. So it's going to be methylation of BRCA1, which will play a role in sporadic breast cancer. So just to conclude about this lecture, which I understand is very complex and lots of information, but all of the different genetic alterations that you can find in cancer genes, they're all related to the all hallmarks of cancer. And I think my colleague last week, Tony Madrick, have introduced to you the solid tumors and hallmarks of cancer. So all of the genes, I'll give you examples like P53, RB, MYC, RB1, they are all involved in these biological processes. So that's really important that you put the genetic changes in the context of biological processes. And some additional reading that if you're interested in genetic changes. There is this Bovary 100-year celebration. Remember, I told you that he published his first somatic mutation theory in 1950. So we are celebrating the 100 year of his